we basically work on aspects related to DNA repair and DNA replication in vertebrate cells. This is highly significant for understanding how cancer develops and how cancer basically arises in humans. Basically, what happens is that in cancer cell, it has been found many years ago that DNA is not stable. This means that the replication and the repair of the DNA is somehow different from normal cells. Indeed, if you make a picture of the DNA, and in this case, if you make a picture of the chromosomes contained in a cancer cell, they look really different from human normal cells. Because what happens is that many of these chromosomes look abnormal. There is an increased numbers, or there is a structural abnormality. And this underlines a very important concept that not only genes are mutated in cancer and altered, but also their position on the chromosomes is not like the position that you would find in normal cells. And this is important because if a particular gene loses a particular position on chromosomes, it loses the way it is normally regulated. And in cancer cell, what happens is that sometimes chromosomes fuse together and or break and are repaired with pieces that do not belong to them. Okay, so we are trying to understand what happens that and what are the problems that lead to this kind of DNA instability in cancer cells. And in particular, we are focusing on genes that are commonly mutated in cancer cells, like, for example, BRCA2 or RAD51 gene family. In BRCA2, mutation that leads to partial loss of this gene predisposed to breast cancer. And this is highly frequent because 5% of women affected by breast cancer have familiar type of breast cancer, and in this familiar type of breast cancer, BRCA2 is highly mutated. So how do we study BRCA2? BRCA2 is a very large gene and consequently give rise to a very large protein. This gene is essential, meaning that if you completely knock it out, like geneticists would do in order to understand the function, you lose the survival of the cell. This means that cells can are very sensitive to complete loss of BRCA2. But this is a problem to study BRCA2 because if we don't have a cellular model, then we cannot delete BRCA2, otherwise the cell would die. How do we overcome this problem? So we decided to use another system in which BRCA2 is highly homologous to human BRCA2 gene. And in this case, this system relies on Xenopus laevis eggs. So the Xenopus laevis is a vertebrate, is a frog that produces eggs that are very large unicellular basically system, where you can easily deplete using an antibody the protein of interest. And in what we do in the lab, we make an extract out of these eggs and we deplete BRCA2 out of the extract, and we study how DNA is replicated and repaired when this protein is not around. And we recently found that when BRCA2 is not around, the DNA is full of gaps. What does it mean? It means that instead of having a normal helical double-stranded structure, it has pieces of region where the DNA is single-stranded. This means that the DNA is not being perfectly duplicated, and this is the first time that this has been shown. In particular, this is very important because the recent drug, namely the PARP inhibitor, which blocks the action of PARP1, which is an enzyme deputed to the repair of single-strand gaps, has been shown to be very effective in breast cancer. So what we think is happening is that BRCA2 is important to repair single-strand DNA gaps, and when BRCA2 is not around, these gaps accumulate. Now, normally, cells are able to cope with this and to repair these gaps by alternative pathways, and this alternative pathway relies on PARP1. But when you block PARP1, this alternative pathway does not work. Therefore, there is a synergistic effect that kills specifically cells in which BRCA2 doesn't work. So this is a first example whereby knowledge on the DNA repair function has been linked somehow to a drug that inhibits a repair function that can specifically target cancer cells and not normal cells. This is very different from chemotherapy that basically does not discriminate between normal cells and cancer cells. Therefore, the idea for the future is to understand even better how the fine mechanism of DNA repair works in normal cells and in cancer cells, using model systems like Xenopus laevis and other systems like cellular derived from cancer cell lines. In this way, we could understand if other factors contribute to the repair in these specific cancers, and to see if this factor can be selectively targeted in order to build knowledge and to propose strategies to create new drugs that then can be used in the future to selectively kill cancer and non-normal cells. So the idea is to evolve towards a selective treatment of cancer cell, and we know that this is in line with what is called precision medicine, which is basically how is the cancer is going to be treated in the future, because now that we can very rapidly have information about a number of genes mutated in a single patient, we can rapidly know what is the genomic and the genetic background in these patients, so that we can administer selective types of therapies in order to treat this patient with high specificity and less toxicity. Of course, we will need to know which genes need to be targeted, and we will need drugs to specifically target this. This will be a long-term goal, but we have now all the knowledge and all the tools set in order to reach this kind of knowledge. So now we think we understand what cancer is. The problem is now how to treat it, and that's what we are trying to do.